All right, welcome to the deep dive. We're diving into some pretty cool biology today.
Definitely cool, yeah.
And we're going to try to make this one especially clear for any of Mr. Martin's IB biology students listening out there.
Yeah, we know you guys are studying hard for those IB exams.
Yeah, or maybe you're just an IBDP student and English isn't your first language.
Yeah, we want to make sure this makes sense for everyone.
Totally. So we're talking genes today,
Right.
specifically gene mutations and gene editing.
Yeah, this is all about how life changes, how it evolves.
Right, it's that big theme, continuity and change.
Exactly. But we're looking at it way down deep at the molecular level.
Okay, so we're talking about DNA, the instruction manual for making all those proteins that make us, us.
Think of it this way. If DNA is like a recipe, then mutations are like little changes to that recipe.
Oh, I like that analogy.
Yeah, like what if you accidentally put salt instead of sugar?
Yeah, or like left out an ingredient completely.
Exactly. That's kind of what a mutation is, a change in those DNA instructions.
So in this deep dive, we're going to break down the different types of mutations.
Yeah, we'll see what can cause them, what kind of effects they have.
And then we'll get into this really powerful tool, gene editing.
Which is basically like having the ability to go in and deliberately rewrite parts of that DNA recipe.
Okay, so let's start with the basics, different types of mutations. The notes mention base substitution mutations first.
Right. Base substitutions are like typos. Imagine you're typing a word and accidentally hit the wrong key.
Yeah, like they instead of they.
Exactly. In DNA, it's when one base, like A, T, C, or G, gets swapped out for another.
And these are also called point mutations, right?
Yeah, because it's a change at a single point in that whole long DNA sequence.
Okay, so we're substituting one base for another. But are there different types of substitutions?
So there are. You've got transversions, where you swap a purine for a pyrimidine, or vice versa. Purines are adenine and guanine. Pyrimidines are cytosine and thymine.
Got it. So swapping an A for a C or a G for a T, that kind of thing.
Yeah, exactly. Then you've got transitions, where it's purine to purine or pyrimidine to pyrimidine.
So like A to G or C to T.
You got it. And interestingly, transitions are more common than transversions.
Huh, okay. Now what happens when you have one of these base substitutions? Does it always cause a problem?
Well, it depends on what that base change actually does to the protein that's being made. Remember, DNA gets transcribed into messenger RNA, or mRNA, and then the mRNA gets translated into a protein.
Right, and each three-letter word in that mRNA sequence, called a codon, codes for a specific amino acid.
Exactly. So you could have a same-sense mutation, which is also called a silent mutation.
Same sense, so the meaning stays the same.
Precisely. The DNA changes, the mRNA codon changes, but it still codes for the same amino acid.
Whoa, so even though there's a mutation, the protein doesn't change at all.
That's right. It's because the genetic code has some redundancy built in. Meaning multiple codons can code for the same amino acid.
Okay, so let's say we have the DNA sequence, pecocati, TTT, hup6, tecocasi.
That gets transcribed into the mRNA sequence.
And that gives us the amino acid sequence.
Yeah.
Methionine, alanine, cysteine, lysine, alanine, tryptophan. Quite a mouthful.
Now imagine there's a mutation and the DNA changes too. TCT coca.
The mRNA could then become AGTC, TCD, EG, TTTG. But because multiple codons can code for proline.
You could still end up with the same amino acid sequence or very close to it.
Wow, that's amazing.
Yeah.
The cell has a built-in safety net.
In a way, yeah. But sometimes that base substitution does change the amino acid.
And that's called a missense mutation.
Yep. And that can change how the protein folds up, its three-dimensional structure.
And if the shape changes, the protein might not work right.
Exactly. And that can affect the phenotype, the observable characteristics of an organism.
Like how they look or how their body functions.
Exactly.
Yeah.
The classic example is sickle cell anemia.
Ah, sickle cell. Our notes say it's caused by a change from CTT to CAC on the DNA.
Right, which leads to a change from JAG to GUG in the mRNA.
So instead of glutamic acid, you get valentin in the hemoglobin protein.
And that one tiny change has a huge impact on the shape of red blood cells.
Yeah, they end up sickle-shaped, and that causes all sorts of problems.
Now, there's another type of base substitution, the nonsense mutation.
Nonsense. What's so nonsensical about it?
Well, in this case, the base change creates a stop codon.
A stop codon. Like a period at the end of a sentence.
Exactly. It tells the ribosome, the protein-making machinery, to stop reading the mRNA.
So you end up with a shortened protein, a truncated protein.
Right, and that shortened protein is usually non-functional.
Okay, so let's look at an example. We have the sequence. TACCG, TACGGGCG.
which becomes OGBRZUGGGGG, giving us
Yep.
methionine, alanine, and then SNP.
Yeah, that's it. Game over for that protein.
And our notes mention cystic fibrosis as an example of a disease that can be caused by this.
Right. Sometimes nonsense mutations are involved in cystic fibrosis.
Okay, so that covers base substitutions. We've also got these things called SNPs.
SNPs, yeah. Single-nucleotide polymorphisms.
Those are just variations at a single base, right? Right, but they're present in more than 1% of the population.
So they're like common variations.
Exactly. They're all over the place in our DNA.
And a lot of them probably come from these base substitution mutations we've been talking about.
Exactly. They're what make us all unique.
Okay, so we've covered substitutions. Now what about insertions and deletions?
Those are like adding or removing whole words from a recipe or even whole sentences.
Okay, so insertions are when you add one or more nucleotides to the DNA sequence.
And deletions are when you remove one or more nucleotides.
So it's not just about whether you're adding or removing, but how much you're adding or removing.
Exactly. If you add or delete a multiple of 3 nucleotides,
Like 3, 6, 9, and so on.
yeah, then you're adding or removing a whole codon.
Which means you're adding or removing an amino acid from the protein.
Right, so with an insertion, you get a longer protein, and with a deletion, a shorter one.
But the reading frame stays the same.
Yeah, the reading frame is how the DNA sequence is read in those groups of 3.
So you're just adding or removing whole words from the sentence.
And the rest of the sentence still kind of makes sense, even if the meaning changes a bit.
Got it. But what if you add or delete a number that's not a multiple of 3?
That's when you get a frame shift mutation. It's like shifting the whole sentence over by 1 or 2 letters.
So all the words after that are messed up.
Exactly. All the codons downstream of the mutation get read incorrectly.
And you end up with a totally different protein.
Often a non-functional one.
And the earlier in the sequence this happens, the worse it is.
Yeah, the more of the protein gets messed up.
Okay, let's see some examples of this. Huntington's disease, is that a frame shift mutation?
It can be, yeah. It's linked to insertions in the HTT gene.
Specifically, too many CAG repeats.
Right. Normally the HTT gene has a certain number of CAG repeats, but in Huntington's there are way too many.
So instead of maybe 10 to 30 repeats, you might have over 40 or even over 100.
And because CAG codes for the amino acid glutamine,
You end up with a Huntington protein that has a really long stretch of glutamines.
and that makes the protein toxic, which causes damage to brain cells.
What about deletions? Our notes mention the CCR5 gene and HIV resistance.
Right. The CCR5 gene makes a receptor protein on immune cells.
And HIV uses that receptor to get into the cells.
Exactly. But there's this deletion mutation called CCR5 delta 32.
Delta 32.
Yeah, 32 nucleotides are deleted from the gene,
And 32 is not a multiple of 3,
so it's a frame shift mutation.
which means the CCR5 receptor doesn't work properly.
And people with this mutation are resistant to most types of HIV.
Wow, so broken gene can actually be a good thing sometimes.
It's a great example of how complex mutations can be. Sometimes they're harmful, sometimes beneficial.
Okay, so those are the main types of mutations. What causes these mutations to happen in the first place?
Well, sometimes it's just errors during DNA replication.
Replication. That's when the DNA is copied, right?
Yeah, every time a cell divides, the DNA has to be copied.
And the enzyme that does that is DNA polymerase.
Right, DNA polymerase is a pretty amazing molecule.
But it's not perfect, right? It can make mistakes.
Yeah, it can insert the wrong base or add or delete a few nucleotides.
But don't cells have ways to fix those mistakes?
They do. There are these proofreading mechanisms like a spell checker.
But sometimes those mistakes slip through.
And that's when a mutation becomes a permanent part of the DNA.
Okay, what else? Our notes mention something about CG-rich regions.
Right, those are called CPG sites. They're areas in the DNA where you have a lot of cytosine and guanine bases next to each other.
And those are prone to methylation, right?
Yeah, methylation is when a methyl group gets attached to a cytosine base.
And that can make the cytosine more likely to mutate.
Exactly. It can spontaneously change into thymine.
And if that change isn't fixed, it becomes a mutation.
Right, so these CPG sites are kind of hotspots for mutations.
What about those short tandem repeats, the STRs?
Microsatellites. Those are regions of DNA with short repeating sequences.
And those have high mutation rates too.
Yeah, because when DNA polymerase is copying these repetitive regions, it can kind of slip up.
Like it gets confused by all the repeats.
Yeah, it can add or delete a few repeats.
And that causes insertions or deletions.
Exactly. And this is thought to be one way those expanded repeats in Huntington's disease happen.
Okay, so just the normal process of copying DNA can lead to mutations?
But there are also these things called mutagens, which can increase the mutation rate.
Mutagens, like things that cause mutations.
Exactly. And they can be physical or chemical.
So what are some examples of physical mutagens?
Well, there's ionizing radiation like gamma rays, X-rays, UV radiation from the sun.
And alpha and beta particles too, right?
Yeah, all these types of radiation can damage DNA.
Like break the strands of DNA.
Exactly. They can cause single-strand breaks or even double-strand breaks, which are more serious.
What about chemical mutagens?
Those are chemicals that can react with DNA and cause changes.
Like how?
Well, some of them can mimic the normal DNA bases, but pair incorrectly during replication.
So you end up with a base substitution.
Right. Others can wedge themselves between the base pairs, distorting the DNA.
And that can lead to insertions or deletions.
Exactly. Some examples are mustard gas, asbestos, benzene, things like that.
And even some infectious agents like viruses and bacteria can be mutagens.
Yeah, they can cause DNA damage that leads to mutations.
Okay, so lots of things can mess with our DNA. Now, one thing the notes emphasize is that mutations are random.
That's a really important point. We can't predict where or when a mutation will happen.
It's like a roll of the dice.
Yeah, kind of. Although some areas of the genome are more prone to mutations than others.
Like those CPG sites and microsatellites we talked about.
Exactly. But in general, mutations are random events.
And another important point is that there's no way for an organism to deliberately change its DNA to get a specific trait.
Right. Mutations happen, and then natural selection acts on them.
So if a mutation is beneficial, it's more likely to be passed on.
And if it's harmful, it's less likely to be passed on.
Okay, so mutations can happen in different types of cells, right? Somatic cells and germ cells.
Right. Somatic cells are all the cells in your body except for your sperm or egg cells.
And germ cells are the ones that become sperm and eggs.
Exactly. So mutations in somatic cells only affect that cell and its descendants.
So if you get a mutation in a skin cell, it's not going to affect your offspring.
Right. But it could cause problems for you, like cancer.
Okay, what about mutations in germ cells?
Those are the ones that can be passed on to your children.
Because they affect the sperm and eggs.
Exactly. And if a germ cell with a mutation participates in fertilization,
Then the embryo will inherit that mutation.
and all the cells in that embryo will have the mutation.
So germ cell mutations are the ones that drive evolution.
Right. They're the source of new genetic variation.
Okay, so we've talked a lot about how mutations can be bad.
But it's important to remember that they're also essential for life.
Because without mutations, there would be no genetic diversity.
Exactly. And without genetic diversity, there would be no evolution.
So mutations are both a blessing and a curse.
That's a good way to put it. Now, let's talk about gene editing.
This is where we can actually go in and deliberately change the DNA.
It's a really powerful technology with a lot of potential.
And the most well-known gene editing tool is CRISPR-Cas9.
Right. CRISPR-Cas9 is like a pair of molecular scissors.
It can cut DNA at specific locations.
And it's guided to those locations by a guide RNA molecule.
So the guide RNA is like a GPS for the scissors.
Exactly. It tells the Cas9 enzyme where to cut.
But it's not quite that simple, right? There's also this PAM sequence.
Right. The PAM sequence is like a little tag next to the target site.
And the Cas9 enzyme needs to see that PAM sequence before it will cut.
It's like a safety mechanism to make sure it's cutting in the right place.
Okay, so once the DNA is cut, what happens?
The cell tries to repair the break.
And there are two main ways it can do that, right?
Yeah. There's non-homologous end joining, or NHEJ.
And a homology-directed repair, or HDR.
NHEJ is kind of like a quick and dirty repair job.
It just sticks the broken ends back together.
But it can introduce errors like small insertions or deletions.
And those errors can disrupt a gene, essentially knocking it out.
Exactly. HDR is more precise.
It uses a template DNA molecule to guide the repair.
And scientists can provide that template with the desired edit.
So you can either knock out a gene or knock in a new sequence.
It's pretty amazing technology.
Our notes mention an example of using CRISPR to treat sickle cell anemia.
Right. Remember how sickle cell is caused by a mutation in the beta-globin gene?
Yeah, the one that makes hemoglobin.
Well, instead of trying to fix that mutation directly,
Scientists are using CRISPR to target a different gene called BCL11A.
BCL11A normally suppresses the production of fetal hemoglobin.
Fetal hemoglobin, that's the type of hemoglobin babies have, right?
Exactly. And it doesn't have the sickle cell mutation.
So if you can increase fetal hemoglobin production,
You can compensate for the defective adult hemoglobin.
and that's what they're doing with CRISPR. They're knocking out the BCL11A gene, so fetal hemoglobin production goes up.
It's a really clever strategy.
And they're doing this in hematopoietic stem cells, right?
Right, HSCs. Those are the cells that give rise to all the blood cells.
So they take HSCs from the patient, edit them with CRISPR, and put them back in.
And the hope is that those edited HSCs will produce healthy red blood cells.
It's really exciting stuff.
Okay, last but not least, we have conserved and highly conserved sequences.
These are sequences of DNA that are very similar across different species.
Or even across very distantly related species in the case of highly conserved sequences.
Like, why would some DNA sequences stay the same for millions of years?
Well, one reason is that those sequences are probably really important for basic cellular functions.
So if they change, the organism might not survive.
Exactly. It's kind of like survival of the fittest sequences.
And another reason might be that those sequences have lower mutation rates.
Maybe because the genes are expressed a lot?
Expressed meaning.
Meaning they're used to make proteins a lot.
And maybe those genes have better DNA repair mechanisms.
So they're less likely to mutate in the first place.
That's really interesting. It's like these are the core instructions for life.
The blueprints that have been passed down through generations.
And they've stayed pretty much the same because they work so well.
Exactly. So to sum up our deep dive into gene mutations and gene editing,
We've looked at the different types of mutations, what causes them, and their effects.
we've seen how mutations are the source of genetic variation, which drives evolution.
And we've explored the incredible potential of gene editing to treat diseases.
We've also touched on the fascinating concept of conserved sequences.
For Mr. Martin's class and all the other IBDP students out there, we hope this deep dive has been helpful.
We know genetics can be a tough topic, but hopefully this has made it a little clearer.
We wish you all the best in your studies. And for everyone else listening, if you enjoyed this deep dive,
Please like, subscribe, and leave a comment.
it really helps us out. Thanks for joining us on the deep dive.